In this next video for Unit 2, we're going to be talking about the ABO, H, and RH blood groups, some nomenclature that's associated with the RH blood group, and subgroups of these blood groups. This is a lot of information, so this is probably going to be one of the longer videos. Okay, so just a little bit of historical information on the ABO blood group system. So in 1900, this is considered the beginning of modern blood banking and transfusion medicine by Carl Landsteiner, he was the one who recognized different patterns of agglutination when human blood samples were mixed in random pairings. So this is when the blood groups were defined as A, B, O, and AB. So he noted the presence of agglutinating antibodies in the serum of individuals who lacked the corresponding ABO antigen. So this phenomenon is known as Landsteiner's law. So it states that normal people possess ABO antibodies to the ABO blood group antigens lacking on their red cells. So this is a little contradictory to some of the information that we talked about in I think it was Unit 1, where we said that people build antibodies either through pregnancy or receiving blood products, or through transfusions. But this is the ABO and antibodies are kind of an exception to that law. These are what are referred to as inherited or naturally occurring antibodies. So those ABO antigens are detectable at 5 to 6 weeks in utero with newborns possessing fewer antigen copies per red cell and less developed antigen structures than adults. Cord blood samples might demonstrate weaker ABO phenotype reactions. And the cord blood sample is what we use to determine the baby's blood type after the baby's been born. So individuals normally produce antibodies directed against the A and or the B antigens that are absent from their red cells. So these antibodies have been described as naturally occurring because they're they're produced without any exposure to red cells. So they're thought to develop through some type of environmental exposure. Full titer of those naturally occurring antibodies happens between 5 and 10 years of age, and they're mostly going to be of the IgM class. So just to talk about that naturally occurring antibodies again for just a second. So for an example, type A person has the A antigen on their red cell, meaning that they lack the B antigen, so they would naturally produce antibodies to the B antigen. Okay, and here's a picture or a visual of a patient's phenotype. So what is their blood type? This is their red cell, what antigens they have on the surface of their red cell. So the type AB person has the A and the B antigens present on the red cell. So they have no antibodies because there's nothing that they can produce an antibody against within the ABO blood group system. So a type A person has the A antigen, so they naturally produce anti-B antibodies, and so on. Okay, inheritance of the ABO blood group. So like most blood group systems, ABO is codominant in expression. So there's one position or locus on each of chromosome 9 that's occupied by either an A, a B, or an O gene. The O gene is referred to as an amorph, meaning that there is no detectable antigen produced in response to the inheritance of this gene. So the group O phenotype is an autosomal recessive trait with the inheritance of 2 non-functional O genes. So designations of group A and B blood types refers to the phenotype, And again, phenotype is kind of the outward or physical expression of the genotype. So the genotype, examples of genotype would be AA, BB, OO. So for a group O person, the phenotype and the genotype are the same because the person would have to be homozygous for the O gene. Remember, because O is recessive, so we have to have 2 copies of it, meaning homozygous, for that person to phenotype as a type O person. So a person who has a phenotype A can have a genotype as AA or AO. A type B person would have a genotype of either BB or BO. So serologically, it is not possible to determine the genotype from the phenotype of an A or B person. So you would have to do the family studies or molecular assays. A type AB person, again, their phenotype and genotype are the same because they've inherited both the A and the B genes. And here's some different variations. So this includes subgroups. So that's what that's why you see the A1 and the A2, which we'll get into the subgroups at the end of this lecture. But here's the different genotypes, all the different genotypes that can be inherited within the ABO blood group system, and then how each of those genotypes would present as the phenotype. And there is a Moodle video on ABO and RH genetics if you want to take a look at it. Formation of the A, B, and H antigens results from interaction of genes at 3 separate loci. So you've got the ABO, the H, and the SE. So H and SE genes are not part of the ABO system, but their inheritance influences the expression of the A and B antigens. So the H gene must be inherited to form those ABO antigens on the red cells, and the SE gene has to be inherited to form ABO antigens in secretions, so such as your plasma and saliva. So the H antigen is a precursor structure. So I always think of it as like a building block. So maybe we're like building a building, and the H is the foundation. So it's the precursor structure on which A and B antigens are made. So inheritance of the H gene results in the formation of the H antigen. So what happens in type A or B individuals is that they convert that H antigen to either A or B antigen. So you can think of this red cell starts out initially covered in H antigens, and through a particular process, all of those H antigens are going to get converted to either A or B. Type O people do not convert the H antigen. So since there's no conversion of that H antigen in type O people, a lot of H antigens are going to remain on the surface of the red cells in type O individuals. So because of that, they would react with anti-H reagent in testing. Type A, B, or O individuals could be either genotype big H, or big H, little h. So individuals that genotype as little h, this is referred to as the Bombay phenotype. So these people lack a functional H gene, and they also lack any ABO expression on the red cells because they don't have those big H antigens to convert to A or B. So the patient is going to forward type. They're going to look like a type O person, but they're really not. This phenomenon is extremely rare, potentially dangerous, because this person would react with type O blood, like in the cases of an emergency transfusion, so they can only receive blood from another Bombay person because their blood will have anti-H. Because these Bombay phenotype patients lack all antigens, including that H antigen, their red cells are not going to react with reagent antisera. So they're, like I said just a minute ago, they're going to look like they're a type O person because you're not going to get any reactivity with the antisera in the forward type, but their serum is going to contain all of the antibodies. So anti-A, anti-B, anti-A comma B, and anti-H. So that would be picked up in the reverse type. So you would have a forward type and a reverse type that don't match, that show a discrepancy. So that would be one of the first indicators that you might have a Bombay type person. So inheritance of the secretor gene, so that's the SE. So they can be inherited as either big SE, or big SE, little SE. This happens on chromosome 19. So this determines whether ABH soluble substances will be secreted. So will these people, will the person have the ABH, will those antigens or the products of them be detectable in body fluids? So these people are referred to as secretors, and it refers only to the secretion of A, B, and H soluble antigens in that patient's body fluids. About 80% of the population are secretors. So in that case, again, A, B, and H substances are secreted. So, and that depends on the person's blood type, what substances they will secrete. So type A will have A and H, a type B will have B and H, type O person will have only H antigen secretions, and then an AB person will have A, B, and H. So if the person inherits little se genotype, they're referred to as non-secretors, meaning that they're going to have neither soluble H antigens or A and B antigens present in their body fluids. And then the Bombay phenotypes are always going to be non-secretors. So they don't have an A, B, or H substance in their saliva because they don't have those A, B, or H antigens. They could have A or B enzymes in their serum depending on their ABO genotype. The blood bank guy has a really good video on the H antigen, so I would suggest taking a look at that. Okay, now talking about the Rh blood group system. So it is located on chromosome 1. It's the most clinically significant blood group after the ABO blood group, and it has the ability to cause hemolytic disease of the fetus and newborn. Rh blood group system is highly antigenic, and again, that just means that it stimulates production of antibodies. It's well developed at birth. It was discovered in 1939 by Landsteiner and Weiner. They reported an antibody made from cells stimulated by the rhesus monkey, which is where the Rh blood group system got its name, from that Rh. 85% of individuals tested agglutinated, 15% did not, so patients were characterized as either Rh positive or Rh negative. So that's where you get the positive or negative part of your blood type. So antigens in the Rh blood group system include, or the ones that we're going to talk about the most are big C, little c, D, big E, little e, and then there are 40 other antigens that we will not get into. So again, Rh blood group system is a pretty large blood group system. The absence of a D antigen does not correspond with the presence of an antibody in the plasma like we have with those naturally occurring ABO antigens. So antibody production for the Rh blood group system has to be stimulated by exposure during pregnancy or transfusion. One exception to that is there might be some naturally occurring anti-big E So some different RH terminologies. So there's 4 terminologies used in the RH blood group system. The first one is called Fisher-Race or CDE terminology. It's based on genetic theories of RH inheritance. So it names the antibody found in the patient's specimen. So Fisher and Race thought that the RH system antigens were inherited as a gene complex, which is a haplotype. So haplotype is a linked set of genes inherited together because of their proximity on a chromosome. So Fisher and Race thought that the RH system antigens were inherited as that complex that codes for 3 closely linked set of alleles. So they said that the D gene is inherited at the first locus, big C or little c is inherited at the second locus, and then big E or little e is inherited at the third locus, with each parent contributing 1 set of genes. So in this Fisher-Race terminology, patients can phenotype as any of these phenotypes that you see here. So if a patient is phenotyped, meaning that they are tested against antisera. So their patient's red cells are tested against commercially prepared reagent antisera. So those antisera, if the patient's phenotyped and they're found to be positive for D, big C, negative for big E, positive for little c, positive for little e, their phenotype would be written as D, big C, little c, E. So anything that they're positive for, so any of these antisera that we're testing against, and you can see what the patient is positive for, you're writing out their phenotype as those positive reactions. But the genotype for this same patient, you have to remember, because and there's a little slash mark separating the 2 possible inheritance patterns, because remember, you have to get one for mom and one for dad. So this phenotype would translate as this genotype. RH negative just means that the patient lacks the D antigen, and there's no such thing as a little D antigen, but sometimes you'll see that little d used kind of as a placeholder to represent that the patient does not have the D antigen. Okay, the next terminology was the Winer, also known as the RHHR terminology. So his theory was that alleles at 1 gene locus were responsible for expression of the RH blood group system antigen on the red cells. So he said that each parent contributes 1 RH gene, and it's either heterozygous or homozygous. He said that there are 8 alleles that exist at that RH gene locus, and this is how he kind of defined them. Those gene encodes a structure on the red cell called an agglutinogen, which is identified with the same antisera. So with your anti-D, anti-big C, anti-little c, anti-big E, anti-little e to identify the antigens. So the main difference in the Winer and the Fisher-Race theories is the inheritance of the RH blood group system on a single gene locus versus 3 separate genes. And then so that's the difference in like the theories behind those. But then there's also differences in how those are written out. So using the Fisher-Race idea here, that these are the alleles that exist, that gets translated into, so you've got the Winer terminology here, how it translates into Fisher-Race. There's little kind of a cheat sheet here. So for example, if you're seeing a big R, that indicates a big D. So you've got a big R here, translates to a big D over here. Then you've got a 0 indicates that the C and the E are little, and so on. So I'll let you look at that and kind of think about that and translate that. The blood bank guy also has a video that does the video is called Week in the D's, but you can start the video at 6 minutes 30 seconds. He does a shortcut kind of to this nomenclature system that is really easy to understand. So if you're having trouble translating this and understanding this, maybe watch the blood bank guy video. He may be able to explain it in a way that you understand a little bit better. Okay, so and then along those same lines, if the patient does not have a big C or a little e, or, I'm sorry, a big C or a big E, then that means that there has to be a little c and a little e. And that's where you use like the 1, 2, Z, and 0 are used with the big R, the 1', 2', Y, and no superscript are used with the little r. So it's just a matter of learning which of these symbols kind of go together. And once you get the hang of it, it is confusing at first. Once you get the hang of it, it's kind of fun. I mean, it's kind of like working a puzzle to translate from one to the other. Okay, another terminology was called the Rosenfield is a numeric terminology, describes the presence or absence of the Rh antigen. This terminology was developed to communicate phenotypic information for computerized data entry. So it doesn't address genetic information at all. So each antigen has a number that corresponds to the order of its assignment in the Rh blood group system. So a red cell's phenotype is expressed by Rh followed by a colon and then a number corresponding to the tested antigen. So if a sample is negative for the antigen test, a minus symbol is written before the number. So you can kind of see an example of that here. But again, this is for the purposes of to use with computer data entry. So this is not something that we would use in the lab. And then the last terminology is called ISBT, which evolved because of a need for a universal language compatible with computers. So again, you've got a numerical code that's assigned to each blood group specificity. We really won't go into this, either one of these. We really won't use either one of these just because they're intended for computer systems. So it's not really that relevant to the testing that we're doing. Okay, frequency of Rh antigens. So you can see the frequency of Caucasians and African Americans of the different antigens. This can come in really handy, and usually the lab that you work in blood bank, they're going to have a big chart. We've got one on the wall in the classroom that breaks it down even further between races or ethnicities, the prevalence of an antigen in the different races. That can come in really handy if you're trying to determine an antibody. Say you're doing a screen on a patient, you've identified that they have an antibody, but you don't, you're having trouble figuring out what that antibody is. These frequency charts can help you determine that, because if they, say for instance, okay, the little e antigen, 98% of Caucasians have this antigen, so the chances of a Caucasian patient having an anti-little e antibody is almost none. I mean, it's very, very rare, because the majority of the Caucasian population has the antigen, and you cannot produce an antibody for an antigen that you possess on your red cells. So these charts can come in handy when you're trying to determine frequencies. And then here's just the most probable phenotypes using that terminology. So that can help you a little bit. I mean, it's taking this same information, but just putting it into the terminology that we just looked at. Okay, now we're going to talk a little bit about subgroups. So we know about the ABO and RH blood groups, but there are subtypes of those antigens as well. So group A individuals, they have the A antigen. They can be either genotype AA or AO. They're going to phenotype as type A. They're going to have naturally occurring anti-B antibodies. So the terminal sugar for the A antigen molecule is N-acetylgalactosamine. Anti-A reacts with A1 and A2 cells. So A1 and A2 are subgroups. We have reagent cells made for each of the subgroups. But anti-A1 reacts only with A1. So again, if you're getting some weird results in your testing, it could be an indication maybe that you're dealing with a patient with a subgroup. Dolichos biflorus is the lectin used to produce the anti-A1 reagent. Helix hortensis is the snail that we use to produce the anti-A reagent. And you will need to know the terminal sugar, and you will need to know the lectin and the snail name. So the A subgroups. So group A red cells that react with both anti-A and anti-A1 are going to be classified as A1, but those that react with anti-A and not anti-A1 are going to be classified as A2. A subgroups are more common than B subgroups. Cells of about 80% of all group A or group AB individuals are A1 or A1B in the circumstance that you've got a AB individual. The remaining 20% are A2 or A2B or weaker subgroups. So the differentiation between that A1 and A2 phenotype is determined serologically using that anti-A1 lectin. So that's that reagent I mentioned just a minute ago that's made from the Dolichos biflorus. Lectins are seed extracts that agglutinate human cells. Anti-A1 lectin agglutinates A1 or A1B cells, but does not agglutinate A2 or A2B cells. So again, taking that reasoning and using your reagents, you can determine if a patient has an A subgroup. Okay, so the subgroups that are possible, A1, which makes up about 80% of your group A people. So the antigen that they produce would be A1. The possible genotypes could be A1A1, A1A2, or A1O. They're still going to produce anti-B antibodies, and they're still going to phenotype as A. It's just going to be the A1 subtype. So with the subgroup A2, which is structurally and genetically different than A1, this person has fewer antigenic sites than an A1 person. So there's also that quantitative difference, about 20% of group A people versus 80% that are A1. So the subgroup A2 person would have antigen A2 on their red cells. They could be genotype A2O or A2A2. They're still going to produce anti-B antibody. Some of them can also produce anti-A1, and then their phenotype is going to be A2. Okay, with the B blood group, so again, the patient has the B antigen on their red cell. They can genotype as BB or BO. Phenotypes that are possible are type B or acquired B. So acquired B is a phenotype, this is actually a type A person who has been transformed to B either through by cancer or bacterial infection. So this is going to show in the forward and back type as a 4+ reaction like a type A, but they're also going to have a weak reaction with anti-B reagent in the forward type due to that B-like antigen that patient has acquired. And it's a lot of times it's from bacterial enzymes that modify the immunodominant sugar. Okay, and then the BA phenotype is truly a type B person who acquires reactivity with anti-A typing sera. So in these people, the B gene transfers the immunodominant sugar for the B antigen and trace amounts of the immunodominant sugar for the A antigen. So a type B person is going to produce anti-A antibodies. The terminal sugar for the B antigen molecule is D-galactose. And then your subgroups of B, they're very rare. You're not going to see those like you will with the A subgroup. So, and they're usually recognized in your testing if you're having variations in your reaction strengths using anti-B and anti-A, B reagents. This is kind of the same information like what we've been going through for A and B. So I'm going to let you go through this. Terminal sugar, D-galactose. So that was an A1B. So this is a person who's going to phenotype as an AB, but they can have A1. Here's the A2. Okay, group O. So the people who type as group O, they have no antigens. And technically, they do have the H antigen on their red cell because remember, the H antigen is that precursor that gets converted to either A or B, but that has not happened in type O people. There was not that conversion of the H antigen. So they're called a type O person, but technically they still have a lot of those H antigens present on their red cell, but just be aware there is no O antigen. So their genotype has to be OO because the O is recessive, so it has to have been inherited from both parents for them to phenotype as type O. And then they're naturally going to produce anti-A, anti-B, and anti-A common B antibodies, no subgroups. Okay, and then with the RH or the D antigen, the genotype, so the RH blood group system includes big D, little d, and again, remember little d is not really a real thing. We just kind of use that as a placeholder to represent the lack of the D antigen, whereas big C, big E, little e, those are antigens. So you've got an individual inheriting a set of Rh genes from each parent as a haplotype. So the pairing of the maternal and paternal haplotype determines the offspring's genotype. So again, they've inherited, say this is what they inherited from mom, this is what they inherited from dad. It's divided by that slash mark to indicate that this is a genotype, but then how would this genotype look as a phenotype? So you have to take that into consideration. So the phenotypes are either the patient's Rh positive or negative. They can produce anti-D antibodies, which is an IgG class of antibody, but that is, these are not naturally occurring, so that's through transfusion or pregnancy. D is very immunogenic blood group system, so you can get some negative reactions. Okay, so some variations on the D. The first one is called a weak D. So this is a weak form of the D antigen. So for us in our blood bank testing, that means that we're going to get a weak agglutination reaction. So it would require additional testing for the detection. Initially, you're going to get a weak D reaction, so a weak agglutination reaction. which indicates that you might have a person who is a weak D. So it can result from a weaker genetic expression of the D gene. That can be due to either position effect, which means that the D gene is inherited in trans versus cis. So trans means it was inherited on opposite chromosomes, or cis meaning that it was inherited on the same chromosome. So if they're inherited on different chromosomes, you're going to have a weaker expression of the D antigen. Or this can also be due to what's referred to as a partial D phenotype. It used to be referred to as a D variant or D mosaic, which means that part of the D antigen is missing, like part of the antigenic structure is missing. So you can kind of think of it like a piece of pie, and a piece of the pie is missing. So weak D testing is part of prenatal evaluations, and Rh immune globulin, also referred to as RhoGAM, which is given to moms to help prevent reactions with baby. It'll give stronger reactions with AHG testing and can be significant in blood donation testing, because the individual is going to appear as Rh negative, but they're actually going to be positive. So, so again, this is if part of that, if a patient is weak D, they're going to initially appear as being D negative. But they're truly D positive. So this is most important when determining baby's blood type and when testing donor units. Because if that donor unit, if that donor was a weak D person, they're initially going to look like they're D negative, but they're actually D positive, and that blood needs to be labeled as D positive so that it only goes to another D positive person. Okay, partial D, like I mentioned, it's sometimes referred to as mosaic. They're missing part of that D antigen complex, so they can make an antibody to the part of the D antigen that they're missing. So partial D phenotype would be suspected if a D positive individual makes an antibody that reacts with D positive cells, but is non-reactive with his or her own cells. So this is not very common. Rh null, so this phenotype appears to lack Rh antigens, and it results from a type of membrane abnormality. So either a regulator gene or an amorph gene is able to produce Rh antibodies. And then some of the other D subgroups are D deletion phenotype, the Del phenotype, Rh mod phenotype, but we're not going to talk about those. Okay, anti-H, Ulex europaeus is the lectin used to create or to produce the anti-H reagent that we use. It also can be recovered from Bombay individuals, because remember, Bombay people do not have that H antigen, so it's represented by the little h. And then because of that, they can produce anti-H antibodies. So anti-H reagent will react with, so this is strongest reactivity to least reactivity. It's going to react strongest with O individuals. So O cells, because remember, those folks have the most number of H antigens present on their red cells, because those H antigens have not been converted to A or B. So anti-H reagent will react most strongly with O cells, then with A2, then with B, A2B, A1, and then A1B. and you will have to know the order of reactivity there also. And then the terminal sugar for the H antigen molecule is L-fucosyl transferase. And then again, just a little bit of frequency of blood groups by ethnicity. So O is the most common blood type in all, well, of the three ethnicities that we're looking at here. So 45%, 47% O positive in African Americans, 39 O positive in Asians, 37 in Caucasians. Then A positive, A positive, A positive, and B positive. So the frequencies are the same in each of the races here. It's just different percentages that you might see. Okay, that's all for this lecture video. Next one, in the next lecture, we'll be talking about testing that we would do for ABO.